Okay, so for your third exam and your final exam, they're going to be completely online. And so what we're going to do in terms of covering the content is that there's going to be a number of videos like this that go over the topics and explain them. Several of them I've already made and cover the first couple topics on DNA replication, gene expression, and so forth. And then there's some other topics that I still have to make up some videos for. So what you're going to have for exam 3 in terms of content in order to study is, first off, you're going to have lecture videos. Like these. And so this one, I'm not going to really give content. I'm just going to give you the overview. So when you're preparing for exam 3, you really need to look at this first to know what it is you're supposed to be doing, where you should find it, and so on. I'm going to try to explain all those things. So the lecture videos, I'm going to make a list of all the topics, and there'll be a little link, and there'll be a little video after that. Similar to ones you've already seen from some of the other topics I've done in the class, but this is going to include now every single thing that's going to be on the exam will be covered in these. And you really need to look at them because this is going to be the primary actual lecture content you get. The rest of it's going to be all on your own. Well, so except from the PowerPoints. So you're also going to have PowerPoints. Okay, so the PowerPoints are going to be a little different. So I'm going to modify them, the ones that I currently have online. So the ones I currently have there have all the content that you need. And you can use those, but I'm going to try to, number one, fine-tune them so that they only include just the material that you need to know, because I'm not going to be able to just constantly give you that feedback every day in the lecture room. Since I'm just doing the videos on the specific topics, there'll be other things in the PowerPoints that kind of, you'll wonder what should you do with those. And this is by reorganizing them, you'll be able to figure that out. In addition to that, I'm going to attempt to see, I've done these before, is a voiceover PowerPoint. Okay, so I'll have the static PowerPoints in Blackboard as they currently are, but then in addition to that, there'll be separate files in a separate folder that have voiceover PowerPoints is what it'll be called. And in those, it'll be a video. But in those videos, instead of seeing me, you'll just see the PowerPoint, and I will just be talking. It'll be a screen capture of my screen, and we'll go just from slide to slide to slide, and I'll narrate through them and essentially say, you need to draw this, you need to be able to explain that, or don't worry about this. This is just extra information. It's interesting, so read it if you want, but don't particularly focus on it. So the voiceovers will be more of a guide to the PowerPoint, and between the two of these, you'll really get 100% of the information. But on top of that, right, you'll also have readings. Okay, so the readings will be assigned. Now, there's not a specific assessment of whether you read it or not, except that I'm going to give you the links to our OpenStax microbiology textbook and the other free online textbook that we're using, and then you should really read those pages because again, this is going to be the main way you're going to get the content in the course. In addition to that, those would be places that you could look up information as you do the online quizzes. So we'll continue with the quizzes. There'll still be quizzes for exam 3, quizzes for exam 4, just like the ones you've already done for the exams 1 and 2. And if there's information you need and you can't quick ask me a question about it, you should look it up on your own and use the reading pages that I assign or the topics that I assign to do that. So that's going to be the bulk of how you're going to find your material. Like I said, there'll still be the quizzes. And then eventually we'll have the exam, and I'll come back eventually with a little review video beforehand, and I'll give you all the details on the dates that'll be available and how you're supposed to do it online and everything else. Now, in terms of the topics, what is on the third exam? All right, so we're going to do an overview of this, and I'm going to kind of tell you which topics I already have videos previously made of and which ones are going to be new and how it's going to work. So we're talking about microbial genetics. That is our main topic for exam 3. So we have to cover DNA, structure, and I have already a whole bunch of videos on the structure of DNA, individual nucleotides. You should be able to sketch one. You should show how nucleotides are going to be able to be attached to one another. And then the overall double-stranded structure of DNA. how that works, the N, 3', 5', being able to label them, and so forth. Right? And obviously answer questions on them. The exams are going to be mostly multiple choice, but I'm working on an option where you will do a few sketches and then scan or photograph them and then send them to me. So I'm going to see how that works. So still be focused on doing that material as well. So you're going to go through DNA structure. As far as replication goes. And then we'll specifically talk about genes. And then the promoter for genes, which are going to be sequences at the beginning of a gene that are responsible for whether that gene is able to be read by the mechanism of the cell and then expressed, meaning that RNA will be made from it, and that RNA will be used in some way to make a protein that has some activity in the cell that is directed by the code in the DNA. So you're going to need to be able to go through all that. But promoters is an additional topic that kind of comes a little bit later on after this. So because we're going to have a topic called regulation of gene expression. Regulation of gene expression. And that's going to come later, so sort of toward the end. But it's going to be related to the genes and the DNA and the promoters, so you really need to know that stuff first. Then we're going to go into the process of gene expression, which really includes two major processes. There's transcription, and transcription is the process of where the DNA is copied into a piece of RNA. So we're going to go over RNA, differences between DNA and RNA. You're going to need to know that. And then we're going to go into translation, which is where the RNA is used, and there's many types of RNA. You need to know the messenger, the ribosomal, the transfer RNA, how they're structured, and so forth, into a polypeptide / protein, and how that protein is, eventually gets to its active form and it does what it does. Now I have lectures already prepared for all these. I don't know, it's about 8 of them or something like that. Okay, so I'm going to list the topics one by one, and I'm going to list those videos. You're going to need to go through those and know that material pretty well. This is what in lecture I would usually go through pretty fast because you should have already seen most of these things before. But now it's going to be more self-paced. You can spend as much time or as little time as you want with it. If it's very familiar to you, then just kind of skim it. If it's something you haven't seen before or very unfamiliar with, then you're going to need to spend more time and rewatch some of those videos because there's a lot to it. So make sure you get through this part. That's just the first part of it. That's just almost like the introduction to what we are really focused on. This is going to be happening before. Now, in our last material, we talked about binary fission. A bacterial cell divides into 2 new cells. Okay, so these are new cells. That's how the material kind of ties together. Before that happened, if you remember, I mentioned briefly the DNA has to double, right? The replication process. We have to double the DNA. That means all the genes in there would be the same, which means all the information would be the same, which means the cells could do all the exact same things, and they would be clones of one another. They'd be exactly the same. So what we want to get into in this exam, which is how we get some genetic diversity. And I will get some new markers. These seem to be dying, and I can't find new ones right now. Genetic diversity in bacteria. All right, so this is going to be a big part then of the exam material, all right? And that's going to be all sort of new, and most of these I have to make up the videos for. I have not made them yet. So what we're going to be looking at here are mutations, different types of mutations, how mutations then affect the outcome, of these new cells, whether they are genetically different or not, what types of mutations can be passed on to other cells, and what types of mutations are not, and so on. So there'll be some just definitions, just straight terminology, and some process that you're going to need to know. They're going to be related to the mechanism of DNA replication. So if you don't yet understand replication, you're not going to get this sort of material. Okay. Beyond that, we're going to have horizontal gene transfer. right, so for horizontal gene transfer, this is where one bacterial cell will be able to pass on genetic information to a different bacterial cell. So this is cell A, let's say this is cell B. And then some new information will be passed into this cell, and this cell will now change. So it will acquire new genetic information, increasing the diversity. And so there are going to be a couple mechanisms that I'm going to go through. One is conjugation, so we'll come back to, again, structure of the cell. The pilus, if you remember before, one cell will actually produce a structure that will connect it to another cell, and then through that structure they're going to pass DNA. So we're going to have to come back to DNA replication once again, and we're going to have a special process here called rolling circle, ing, replication. So it's going to be a unique type of DNA replication that's going to occur in what we're going to call F+ type cells that are going to make DNA that's as a single strand that's going to be passed through the pilus and go into the new recipient cell. So this is going to be a whole process we're going to go through called conjugation, and there are going to be other processes where cells are going to get new DNA from a virus. So a viral infection is going to transfer genetic information into the cell, and the cell is going to keep that information and will forever be changed. New genetic information because of that infection. And then you're also going to get pieces of DNA taken up from the environment, which is another type of a transformation process. So there's going to be a couple different processes. This is going to be a big part of the information as well. Lastly, there's one other thing we're going to cover, and that is going to be then, it's kind of a little more complex topic, will be back to the regulation of gene expression. We're going to kind of come back to that now. So this is going to be, we're going to talk about operons in bacteria, and how for a bacterial genome, what can happen is that you can have regions like a promoter, which is usually for a single gene, that control the expression of multiple genes all at once. So bacteria are going to have a small genome, and they're going to have to kind of conserve that information. So what's going to happen is they'll have these larger areas, or these areas that can control larger expression of genes all at once, as opposed to all of them separately. They can kind of combine them. So that's going to be a process that we're going to go through. There'll be terminology again, and it's all sort of connected. And that's going to be the bulk of the material that's on your third exam. So it's all microbial genetics. The first part's just DNA, RNA, transcription, translation. Then we're going to get into how diversity is created in bacteria and go over some of those processes that can establish it. And then finally, we'll look at then how differential gene expression and control and regulation of gene expression can also affect organisms. So for example, like in our lab, the one thing you've seen is there's a particular bacteria that sometimes looks pink and sometimes doesn't. Serratia marcescens. It has a heat-dependent gene expression. So it has a certain gene to produce a pink pigment, but it doesn't always do that. So this is just the overview of material for exam 3. Now, after watching this video, you should go into the list of topics and then start watching those videos in order. Once you get to the point where you've covered all the topics, to do, say, the first quiz, and you'll take the next quiz and so forth, work your way through. PowerPoints, these are going to be new ones I'm going to have to make with voiceovers. So look for those. There'll be a new special folder that just says Voiceover Powerpoints, and those will have the updated PowerPoints with hopefully a narration for them, so you can see that. And then just check out the assigned readings. Those will be just links I put in there in your textbooks that you can go to these topics if you want more information or need to look up information. Right, and that should cover what you're going to need to know. I mean, as an overview, not the actual content, but the overview of what we're going to do for exam 3. And then, like I said, I'll be posting when the date is for that. It's essentially going to be online for a period of time. You'll have about a 1-week period of time where the exam will be online, and you'll be able to do it any time within that week. So, 8 a.m., 8 p.m., midnight, it doesn't matter, but you'll have a certain, once you start it, you can only do it once. The quizzes, which you can repeat, are going to be, same sort of format, but obviously you can repeat them. The exam you will not be able to repeat, and there will be a time limit on your exam, which means once you start it, clock is going to start, and it's going to just tick away. And once it's done, it's going to just automatically submit whatever you've answered. So if you've only answered half the questions, it's just going to submit those, and that's all that will be graded, because I know that it's just impossible to say, oh, don't look up your notes or don't look up resources online. That's what's just going to happen. If you prepare properly, actually study and read and watch the videos, you'll be able to get through all the questions in half the time that I've allotted for you, and have a whole bunch of extra to potentially look up some things and just double check stuff and say, okay, now I answered everything, but I'm not sure about these. Maybe I'll go check them. If you don't study at all and you go ahead and try to look up every single thing, you will never finish. There are going to be a large number of answers that you're not going to be able to complete at all, and they'll be just unscored, or they'll be scored as zeros essentially. So be sure you study as if you're taking a real exam in class, like you would normally study, but now you have a lot more control over the lectures and the time you spend doing it. So I'll address the lab, what we're doing for lab separate. I'll have a separate video on how to complete the lab material, which I'm going to have new stuff online for you, and you're going to have a couple assignments you're going to have to turn into me to kind of make up for what we're not going to do in lab, but for you to still get the basic information from that. And as far as the literature assignments, I'm going to be posting those. I'll give you your grades back for those and give some comments, and then you're just going to be submitting them to me through email. You'll just submit we'll still do that just as planned. I'll just give you new due dates, and you'll just turn them in. And that should be it.